Okay, I will just slowly start now, and maybe some more people will join during the introduction. Yeah, happy Tuesday, everyone. Great that you could make it to today's talk, which is by Elliott Smith from the University of Mexico, as well as the Smithsonian National Museum of Natural History in the US, who will be talking about reconstructing historical sea otter ecology with zooarchaeology and stable isotopes today. So a bit about the format of today's seminar. We will start with the welcome and announcements, which is what I'm doing right now. I'm your host, Nina, by the way. And we'll have Emma's talk, which will last for around 30 minutes. And after that, you'll have the opportunity to ask questions during the moderator's Q&A. More about that later. And then afterwards, Emma's also agreed to stay for tea time. So if you have any very in-depth questions that you would like to ask, or if you just want to have a chat about her workplace or something else, you're also very free to do so. So before we get to the topic of today's talk, a bit of housekeeping. So PaleoPerks values the participation of all folks interested in the paleosciences. And in lieu of that, please remember to abide by our code of conduct during today's seminar, which you should have signed if you are here. Also, please keep yourself muted during the talk out of respect for the speaker. Then you can also ask questions by chatting to the questions at PaleoPerks host, which today will be me, I think. There's no questions host. You can either send the questions directly to the chat, or you can send them to me, and then you can ask them anonymously, or you can also use the raise hand function, and we will unmute you. We also have closed captions built into Zoom, and you can use the CC button that will either show or hide them. Finally, we're always looking for new speakers. PaleoPerks only works because we have so many cool early career scientists that want to give a talk here. So if you know someone who could give a cool talk, or maybe if you want to give a talk yourself, because we also like to have self-nominations, you can nominate someone on our website or on the link here. And finally, we also collect weekly feedback data after each talk. This helps us with our demographics to see if we can improve anything. So if you want to fill out this feedback form, which is completely optional and anonymous, we would also really appreciate it. And with that, more about the speaker, Emma Elliott Smith. Emma did her bachelor's in ecology and evolutionary biology at the University of California in Santa Cruz in the US. Then she moved a bit more to the biology side by doing a PhD in biology at the University of New Mexico in the US. And she's currently a postdoc at the University of New Mexico in the US, as well as a research associate at the Smithsonian National Museum of Natural History, also in the US. And we'll hear today what she is doing there. So with that, Emma, I would like to invite you to share your screen and start your presentation.
Absolutely. Thank you so much for that introduction. Let me just get screen sharing set up. All right. Looking good?
That looks great.
Excellent. Okay. Awesome. Well, thank you, Nina, for that lovely introduction. Thank you also to the PaleoPerks Steering Committee for the invitation to speak, and then, of course, everybody who's tuning in with us today. I'm really excited to be sharing some of my past and ongoing work using zooarchaeology to help inform conservation biology. Yeah. I want to first start by acknowledging some of the many people that contributed to this work. The kind of interdisciplinary research I'll be talking about today is only made possible through collaborations, and I feel really privileged to have worked with a kind of unique blend of anthropologists, archaeologists, as well as geochemists and marine biologists. So just a little bit about me and what I do. As Nina mentioned, my formal training is in biology and ecology, but this field has a really big blind spot, which of course is time. No surprise, right? That's why we're all here at this seminar today. And so most of what we know about our modern food webs and species interactions come from studies that have taken place over the last 50 to 75 years. And what this means is we've been studying ecological dynamics in ecosystems that are already heavily impacted by human activities. And this realization is really what pushed me personally into looking farther back in time. Because if we want to understand what's possible, we need records that reach further back than our modern ecological monitoring does. And so that's where zooarchaeology, which is more the focus of this talk, and paleoecology, which I also work a little bit in, really come into play. Because these records essentially are time machines, right? They preserve bones, shells, and tissues that can record the life histories of individual animals and ecosystems. And to connect all of this, I use stable isotope analysis, which in a sense gives us a common chemical currency for comparing the past to the present. I employ two types of isotopic analysis in my work that we will be talking a lot more about in detail. So this includes bulk tissue analysis as well as isotopic analysis of individual amino acids. But at its core, what stable isotope analysis does is it takes advantage of the fact that you or I or sea otters incorporate the chemical signatures of our diet and our environment into our tissues. And so as a result, isotopic values can allow us to reconstruct where organisms were feeding and their position in the food web. So now onto the main subject of this talk, which is sea otters. So I feel incredibly privileged to have worked on this species for almost a decade at this point. But just to give a little primer for those of us that may not be as familiar with them, as is probably obvious from the name, sea otters are a marine mammal, but it might be more appropriate to call them a coastal mammal. So the map up here on the top, that purple band shows their modern geographic distribution. You can see it's really narrowly restricted to a coastal range. Throughout this geographic distribution, they're found in a wide range of coastal habitats. So they're famous for their association with rocky reefs and kelp forests, but they're also found in soft sediment environments such as estuaries and seagrass beds. Within their habitats, they are voracious consumers of coastal macroinvertebrates. You can think of sea otters as the hummingbirds of the sea because they have incredibly high mass-specific metabolic rates, partially due to their lack of insulating blubber that other marine animals like pinnipeds and cetaceans possess. And so because they eat so much, they actually play an outsized role in shaping their ecosystem. So they're what's known as a keystone species, which means that even in very small numbers, sea otters can completely transform the structure of coastal habitats. But the story of sea otters isn't purely ecological, it's also deeply human. And our relationship with these animals is quite complicated. So it starts, of course, with indigenous communities. So indigenous peoples of the Northeast Pacific coast of North America have deeply rooted traditions of sea otter harvest and management. And this stretches back at least 10,000 years, which gives us a fantastic archaeological record to study. These traditions continue today and are part of a broader system of stewardship that ties people, animals, and coastal habitats together. Sea otters were and are hunted for their dense and valuable pelts, and also in areas near village sites, they're hunted to keep them away from disturbing nearshore clam and shellfish gardens. But despite this millennia of harvest by indigenous communities, until the 1600s, sea otters were found nearly contiguously from Japan all the way down to Baja California, as you can see in this yellow band here. But then during the maritime fur trade era of the 1700s and 1800s, their remarkable fur became a global commodity and sea otters were hunted to the brink of extinction. And unfortunately, indigenous communities were often coerced or forced into this trade, hunting sea otters under really violent colonial systems. So by 1900, it's estimated that only 1,000 to 2,000 individuals were left worldwide. And you can see the red circles on this map show the remnant populations that we're aware of that were scattered throughout the Pacific Rim. Then in the mid-1900s, following some landmark legislation in the US and Canada, conservation efforts to bring the species got back underway, and scientists began translocating sea otters from these surviving groups to other parts of their former range, which is denoted here by these black arrows. Some of these reintroductions were remarkably successful, while others never took hold. And then today, bringing back that purple geographic distribution band, you can see that sea otters in Alaska and British Columbia have recovered, for the most part, their pre-fur trade distribution, but big gaps remain. And this patchy recovery raises, for me, a more broad question, which is how are we defining recovery, right? So counting animals and mapping ranges is an important part of the story, but I'm also interested in trying to understand if sea otters have recovered their ecological role. And that's why we need a historical perspective. So by turning to the archaeological record, we can reconstruct what sea otters were doing prior to this industrial exploitation. So we can ask what habitats they were using and how they interacted with coastal ecosystems and by proxy, local indigenous communities. And once we have these critical baseline data, we can start to evaluate whether sea otters have actually recovered into their historical ecological niche. So this sounds great, but now we have to address how on earth can we actually do this? How can we know what an animal was eating 500 or 2,000 years ago? And as I've already alluded to, right, this is possible through isotopic analysis of well-preserved archaeological specimens. So this photo is an example of sea otter femora from an archaeological site in Southeastern Alaska where I've worked. And if the organic material is still present in these bones, then we can use chemical analyses to make inferences into the ecology of the individual this bone ultimately came from. So we're going to start by talking about bulk or whole tissue isotopic analysis. So in this case, we're looking at bone collagen for archaeological specimens, which is the most abundant protein we find in bone. And we're specifically going to be talking about stable carbon and nitrogen isotopes. We typically visualize these data by plotting carbon isotope values on the x-axis and nitrogen isotope values on the y-axis. If you're not familiar, we report isotopic ratios in what's called delta notation. And all this is expressing the relative abundance of heavy and light isotopes in a sample relative to international standards. And the units of this are parts per thousand or per mil. And this biplot can actually be thought of as sort of an ecological map where each axis represents a key dimension of an organism's niche. So for instance, carbon isotope values differ among different types of photosynthetic organisms. So when it comes to sea otters and the habitats they live in, what we find is that phytoplankton tend to have much lower carbon isotope values than co-occurring macroalgae such as kelps or seagrass and eelgrass. And then nitrogen isotope values are generally used to provide a metric for trophic level, given the fact that we see stepwise increase in nitrogen isotope values as you move up a local food chain. And the word local is very important there because baseline isotopic differences among regions can really alter the absolute values of these. So animals that are plotting in different quadrats of this space then you can think of as corresponding to animals that are feeding in different habitat or trophic combinations. And so what I'm going to do today is I'm going to walk us through some isotopic data sets for pre-fur trade sea otters that we have from 3 geographic regions: Southeastern Alaska, Northern Oregon, and Central Southern California. And these regions are really important because they represent a range of sea otter recovery trajectories and history. So we'll start with Southeastern Alaska. These samples come from the ancestral and unceded homes of the Tlingit and Haida peoples. This is an area where translocations were very successful. And so today, sea otter populations are doing really well in this region and have largely recovered. But this has also led to conflict among sea otters and local communities. Next, we'll go to Northern Oregon, the unceded ancestral homelands of the Tillamook and Clatsop peoples. This is a very different region from Southeastern Alaska. So translocations were attempted in Oregon, but they failed. So there are no established sea otter populations in the region today. But there are sort of some grassroots efforts and interest in maybe trying to translocate sea otters to this region in the future. And then finally, Central and Southern California, the sites we'll be looking at come from the ancestral homelands of the Chumash peoples. This is an area where translocations have had mixed success. Some areas, sea otter populations are recovering and doing quite well, and in other areas, they have not recovered yet. All right, so I'll be showing data first from Southeastern Alaska, and I want to draw your attention to this last bullet point here, because this is a really big issue right now in Southeastern Alaska, is there is a lot of concern from local communities and commercial fisheries about the impacts that sea otters might have as they're re-expanding into this area on local livelihoods. So specifically, we're going to be thinking about clams and bivalves, which are an important source of traditional food for local communities, predominantly indigenous communities. And then we'll also be thinking about sea cucumbers and Dungeness crabs, which support commercial fisheries in this region. And so using isotopic analysis of zooarchaeological sea otter specimens might help us forecast what sea otter diets in this region will look like as they continue to re-expand. So here's a zoomed in map of Southeastern Alaska. The purple stars here denote sites where we have sampled archaeological specimens. So this includes 6 sites. That biggest star is where about 24 of our samples come from. So we think we have about 38 individuals represented. They're almost certainly locally harvested animals. Happy to talk about the evidence we have for that in the Q&A. And radiocarbon dates of associated strata suggest that these specimens were all harvested between 1,000 to 2,000 years ago. And then because there are modern populations in this region today, we were able to also do some isotopic analysis of these recovering populations. So the circles here denote 2 sites where we have sampled sea otter populations from today. And then we're also fortunate to have a really large isotopic data set of potential prey items for sea otters in this region. So these have also been sampled from that same island where we have data for modern populations. All right, so here, going back to this biplot, and I've left our little clip arts on there to help remind us what different parts of this graph mean. So here are the prey data, and there's a lot going on here. So in the red circles, what I've highlighted are those invertebrate species that are really important today for local communities or regional commercial fisheries. And here are the data, I'm just throwing it all up there right at once, the archaeological sea otter specimens. So I have corrected these data for temporal, trophic, and tissue effects. Happy to discuss this more later on, but in the interest of time, I'd like to focus on what these data tell us. So generally what we're seeing here is that sea otters in the past were heavily focused on low trophic level prey, particularly filter feeding bivalves, as well as potentially some red sea urchins. We're also noting not a ton of trophic variability, right? So only a few per mil range in nitrogen isotope values. So the next question is how do these data compare to data from modern sea otters in the region today? So looking first at the data from one of the 2 sites, QU Island in the south, this was a really well-established population at the time it was sampled. And what you can see is that there's a shift to the right on this graph, which suggests a greater reliance on red sea urchins. And then that secondary site at the time of sampling, this was an active re-expansion front, so sea otters had kind of just gotten into the area. And we're seeing, again, a shift in this case towards green sea urchins. But generally, with both these modern populations, we're seeing a really heavy focus on sea urchins. And this is a pattern we'd expect for newly recolonizing sea otters in an area where they have been absent for a couple of 100 years. So we're going to be interested to see how these data change. But also what we're noting is a more limited range in the bulk tissue isotopic values of our modern specimens as compared to these archaeological samples. So what do these data suggest more broadly about the future for sea otters and local fisheries and communities in the region? Well, our data really don't indicate that sea otters in this region were heavily focused on either sea cucumbers or Dungeness crabs. So I've highlighted them in red here, and you can see with maybe one exception, we really don't have a lot of evidence that otters were eating large numbers of these species. There are some important caveats to that interpretation that I'm happy to discuss offline, but overwhelmingly our data don't suggest that it was the bulk of their diets. But the story is very, very different when it comes to the potential impact of sea otter populations on the lifeways of local communities. So many households in this region rely on traditional subsistence harvests of clams, particularly butter clams, which is the species shown in this photo here. And these communities have already seen declines in local clam abundance as sea otters have come back into the region. And if sea otters ultimately reestablish back into their historical niche space, our data suggests that this trend is going to continue. And so what these results really underscore for us is the importance of a community-driven approach to managing sea otters in this region so that we can make room for both people and wildlife. All right, so it was a whirlwind tour through Southeastern Alaska. And now we're gonna basically start moving down the coast and making some more regional comparisons. So we'll start with Northern Oregon. And as I mentioned, this is an area where translocation attempts occurred in the 1960s, but these efforts failed. So there are no established populations of sea otters today. You will occasionally see transient individuals coming down from Washington, but no established populations. But there is a lot of renewed interest, driven predominantly by tribal communities in the region, to bring sea otters back to Oregon. But the big question when thinking about translocations is, where do we put these animals to give them the best shot at survival and establishing populations? And advocates for new translocations in Oregon have argued for estuaries and soft sediment habitats. So the map I'm showing here shows just some of the many estuaries that are present in Oregon. And so it's thought that these might be a good place for sea otters. So what we were trying to do is understand whether sea otters were using these habitats in Oregon in the past. So now this is a zoomed in map of sort of Northern Oregon, and our data come from two sites, Palmrose and Partee. These represent a similar time period as the Southeastern Alaska samples. And I also want to point out these pink stars here on the map are located just south of the Columbia River mouth, which is a major estuary in the region. So if we were going to see evidence for estuarine use by sea otters in this area in the past, these are two really good sites to look at. And then because there are no modern sea otters that we can compare our data to today, we started by comparing our isotopic data from Oregon to these data sets from Southeastern Alaska and then also California. So back to our biplot. Here are the data we've already seen for Southeastern Alaska sea otters. And here are the data from Northern Oregon. So there are some really interesting differences among these data and those from Southeastern Alaska. So notably, carbon isotope values for Oregon are on average about a per mil higher than they are for Southeastern Alaska. That is statistically significant. But there's also a really significant shift in the nitrogen isotope values of these two groups. So I have teed you up to think that this could be trophic level, right? But this is actually why I highlighted how important it is to think about local baseline nitrogen isotope values, because we think this is more likely a function of oceanographic differences between these two data sets. So Southeastern Alaska sits within the Alaskan current, whereas Northern Oregon is within the California current. And the California current is a strong upwelling system that brings isotopically heavy inorganic nitrogen to coastal regions. So we think this is more a function of oceanography. And this becomes more evident when you add in data that we have from California. So now you can see that Northern Oregon and Central California have very similar average nitrogen isotope values. And this makes sense since they're both part of the same current system, whereas Southeastern Alaska has a different local oceanography. So now let me take a step back and provide a bit more context for the mass of data I threw up there from California. So these are from my dissertation research, which was published in 2020, and these samples come from 11 archaeological sites that span 350 kilometers and nearly 5,000 years. So there is a lot of really cool pieces to the California story. Unfortunately, I don't have time to talk about it, but if you're interested, please let me know after the talk or shoot me an email, and I'd be happy to send you a copy of our paper. Okay, so now back to the regional comparison. What's really interesting here is this range in carbon isotope values on this plot. So particularly this trend where Oregon and California tend to have higher carbon isotope values than most of these Alaskan specimens. So does this reflect real habitat differences among these groups in the past? It's hard to say with these data alone. And so this is really where this sort of second type of isotopic analysis that I use in my work comes into play. And so in this case, instead of looking at whole tissues, we're going to be looking at isotopic values of the individual molecules that make up those tissues, which, because we're talking about proteins, means we're looking at individual amino acids. I make these measurements via a GC-IRMS system. If anyone's interested in how we do this, feel free to ask me a question after the talk. But today I'll be presenting carbon isotope data from essential amino acids that we've isolated from the same archaeological specimens we've been talking about. And we focus on these compounds for a couple of reasons. So the first is, as the name implies, most heterotrophs can't synthesize essential amino acids themselves. They have to get them from their diet or, in some cases, their gut microbiome. What this means is that organisms are less likely to catabolize these compounds, and instead they tend to route them directly into their tissues. And the result is that there's minimal isotopic fractionation of these compounds with trophic transfer. And you don't have to take my word for it. This is an example of a feeding experiment that was done on killifish to test this idea. So the y-axis here is the isotopic difference between the muscle sample of, in this case, a fish, and its diet that it was fed in the lab. And that red box here, I just want to point out that across different dietary treatments, which are these different symbols, and 5 essential amino acids, we see no isotopic fractionation with trophic transfer. So that's fantastic, because what that really means is that these isotopic values are sort of imprinted at the base of the food web. So the essential amino acids in a sea otter were synthesized by something like a kelp or a phytoplankton. Okay, so then, in order to interpret these data, we need a comparative data set of primary producers from these different habitats that we might find sea otters in. And so I've looked at this by collecting coastal marine algae from a number of different sites. And specifically, I've measured green algae, red algae, kelps, and phytoplankton. I'll be presenting data for 5 essential amino acids, and we use an ordination, in this case, linear discriminant analysis, to help us visualize these multivariate patterns. So boom, here we go. Here's a lot of data all at once. And I've added those familiar clip arts back on to help orient us to the important parts of this graph when it comes to sea otters. So why kelps are on there is probably obvious. And then phytoplankton are really important because sea otters that are living in these sort of estuary and sandy bottom habitats, are eating things like clams or filter feeders, which means essentially they're living in food webs that are fueled predominantly by phytoplankton production. I also want to note that in this data set, we have a series of historical kelp samples. So these are from Smithsonian botany collections. They're highlighted here in these black circles. And I just want to point out that we don't see isotopic differences in this multivariate space through time, which is fantastic because it means we can feel more confident in using this framework to evaluate our archaeological sea otter samples. So we're going to go ahead and plot our sea otters directly onto this space. And remember that we can assume there's little to no isotopic fractionation with trophic levels for these compounds. Okay, so here are the data from Southeastern Alaska, and we see a large proportion of individuals plotting over here by phytoplankton, but also maybe more individuals than we would have expected plotting in that kelp ellipse based just on their bulk tissue data. Northern Oregon looks a little bit different. So now all but 2 individuals are plotting right on top of that ellipse for kelps. And then finally, California mirrors what we see in the bulk tissue data set, where individuals were occupying a wide range of habitats from sort of the kelpiest of the kelpie all the way to individuals that were very clearly living in phytoplankton-fueled environments, probably soft sediment habitats. Okay, so the next step for us was to conduct a bootstrap resampling approach for this model to evaluate the sensitivity of this classification for our sea otter samples. So what we found, shown here in the blue, are individual specimens that consistently classified with phytoplankton, or what is denoted here as pelagic sources, across every bootstrap iteration. So we're confident those are individuals that were living in phytoplankton-fueled environments. And then same thing in gray is shown for sea otters that we're consistently classifying with our kelp samples. And then these checkered boxes represent what we're calling a mixed classification. So these are specimens that switched which group they were classifying with, depending on the randomized data set within the bootstrap model. And so we're interpreting this to mean that these are individuals that were probably getting their essential amino acids from a mix. So maybe they were using a range of habitat types. So overall, if we take a step back, what we found, firstly, we have evidence that kelp forests have been important to sea otters in the past across all 3 geographic regions that we looked at. This makes a lot of sense. This, of course, is the habitat that they're famous for their associations with. But we see that there is variation among regions. So for instance, 15 of our 17 Oregon samples were clearly reliant on kelp forests, whereas ancient sea otters in Alaska and California were using more of a mix. And this is really important for Oregon and particularly because remember that advocates for new translocations in the region have really highlighted these estuaries and soft sediment habitats as critical areas to consider for sea otter translocations. And we, at least in our current data set, don't have a ton of evidence that sea otters were heavily using these environments in the past. One really important caveat is we did only have 2 sites that we have sampled thus far. So we're really working to expand this data set and incorporate a wider range of regions within Oregon so that we can dial in on how widespread this pattern is. But in general, we think that future translocation efforts should probably take this into account. Okay, so to sort of bring us back, I know that was a whirlwind tour through a lot of data, but remember that these were kind of the main questions we were focused on, is what habitats and prey items were sea otters using prior to their near extirpation, and have they recovered? And really, we haven't quite dialed in on that second question yet. Before that first question, we have provided important data sets that suggest that sea otters were living in a wide range of habitats in the past and exhibited overall broad dietary diversity, but that there are really important differences that at least our current data set suggests existed between geographic regions. And so more broadly, now that we have these data, this will hopefully help us characterize whether these modern populations are occupying their historical niche space and provide, we're hoping, critical information for future conservation efforts. So then just to bring it all home, right, all of these insights are made possible because of both the zooarchaeological record, our modern understanding of the species, and these powerful chemical techniques. So to me, this really highlights the power and the importance of interdisciplinary studies as we try to find ways to balance the needs of both people and wildlife on this increasingly dynamic and changing planet. So thank you all so much for listening. I will be happy to take any questions. And I just want to, again, acknowledge the many, many colleagues and collaborators on this project, because this would have been impossible without all of these wonderful people. So thank you so much.
Thank you so much, Emma. That was a really fascinating talk. Quick reminder to everyone that you can send questions either directly in the chat, or you can send them to our questions@PaleoPerks host. So we do actually have a host, Gwyn today. We did already receive a few, so I will just get started with the Q&A.
Absolutely.
Okay, I think this is one that you hinted at you would be willing to answer. How do you know that your samples were locally sourced?
Yes, that's a great question. And that's something we thought a lot about, right? Is how do we know, A, that people weren't traversing long distances to get these samples, and B, that there wasn't trade. And it's impossible to say with 100% confidence, right? So that's an important thing to recognize right off the bat. But what we've done are look at a couple of metrics that suggest to us that mostly these were locally harvested animals. So one of the first things we've looked at are the distribution of the kinds of bones of sea otters that we find in these sites. So what we see in modern indigenous traditional harvest of sea otters is when they're trading, what they're trading are the pelts. And so when they skin sea otters, they leave the phalanges So both front feet and back feet, as well as the tailbone. So if most of the assemblage we were measuring was from trade, we would expect to see a higher than average percent of finger and tail bones relative to things like rib bones, long bones, and skulls. And it's not the opposite, but we see a very normal distribution of skeletal elements in this assemblage. So that's really strong evidence for us that most of these samples come at least not from traded animals. And the other, sort of pragmatic thing to think about with this, right, is that sea otter meat spoils very quickly, right? So if these were samples that were being harvested and being transported, they probably were not being harvested and transported very far because people were hunting these animals via dugout canoes, right? And while they were absolutely and still are experts in coastal maritime technologies, there's really only so far you can get with a dead sea otter before that meat and the pelt are no longer usable. So those are sort of the metrics we've been looking at. And in all of these regions we've assessing, that's also one of the reasons where, when possible, we've been focusing on isotopic analysis of femora and other long bones so that we can reduce the possibility that we're measuring animals that were traded in. So hope that addresses that question.
Thank you. That makes sense. And I think it's really cool that you can answer this question largely based on knowledge of the historical traits. Let's see. We have a question from Maura in the chat asking, thank you for your presentation. Did any paleoecological data on food competitors provide any insight on past ecosystems for otters?
Can you repeat the question, Nina? Apologize.
Yes, sorry, I'll be a bit slower. Did any paleoecological data on food competitors provide any insight on past ecosystems for otters?
Any data on food competitors? So does that mean, are you interested in other animals that would be competing with sea otters for these same macroinvertebrate species?
Yes, that's what she's asking.
Perfect. Okay, great question. Yeah, so there are a couple of species that can compete with sea otters. But it varies across their range. So in Southern California, there's a fish known as the California sheephead. It's actually a really cool species. It switches at a certain size class from being male to being female, or excuse me, from being female to being male. And when they have this switch, they have this crazy morphological change that enables them to consume large quantities of shelly macroinvertebrates. So clams, sea urchins, exactly the same kind of species that sea otters will eat. Unfortunately, sheephead really don't go that far north. They're more of a warm water species, so we don't have them in places, excuse me, like Alaska. The other species that are often considered a sort of competitor to sea otters, or at least that serve a similar ecological role, are large carnivorous sea stars. So like Pycnopodia, the sunflower star, is a famous example. And unfortunately, that's another case where there aren't many parts of sea stars that preserve well in the archaeological record. So within Southern California, we can kind of compare and contrast what we see in California sheephead, which are a bony fish, to sea otters. In farther north, in Oregon and Alaska, we're a little bit limited in the types of competitors we have for comparison, right? And the other, I guess, complicating factor is that some of these other coastal marine mammal species that we do see in archaeological sites across the range, like fur seals, sea lions, that kind of thing, they're mostly piscivorous. And sea otters really don't eat, they can eat fish. It does occur, there are documented cases, but it's really not the bulk of their diet. So in Alaska, we really don't have that kind of data set, unfortunately. And in Southern California, I actually don't know that anyone has explicitly compared sheephead and sea otters from the same archaeological sites. But I know the data exists, so that's a really great follow-up question that we'll have to dive more into.
Thank you. I think that was a very great answer. Maura also says thank you, so I think that answered the question. we have a bit of a technical question. Someone asked, what is GCIRMS and how does it compare to more traditional isotopic methods?
Oh, music to my ears. I could talk about this technical stuff all day. So GCIRMS stands for gas chromatography, isotope ratio, mass spectrometry. So that is the type of method that is, well, one of several methods that is often used to look at compound-specific isotopic patterns. That contrasts with what is the traditional method for bulk tissue analysis, which, when you're talking about carbon and nitrogen isotopes, is EAIRMS, or Elemental Analysis Isotope Ratio Mass Spectrometer. And really all those acronyms mean is that there's a different instrument that is connected to the isotope ratio mass spectrometer. So in both cases, GCIRMS or EAIRMS, we are using the same analytical instrument to measure isotopic ratios. But we have different we call them peripherals, so different components that are connected to the IRMS, and these behave differently for bulk tissue samples versus compound-specific samples. So for bulk tissue samples, basically what we do is a little bit of, in this case, bone collagen gets dropped into a combustion reactor. It's converted into CO2 and N2 gas. And then we use a short gas chromatograph column to separate the CO2 from the N2, which then get introduced separately into the mass spectrometer, where we can analyze the isotopic ratios. For GCIRMS, we kind of do it in an inverse fashion. So we have, I use a 60-meter GC column, so incredibly long. And in this case, the column is used to separate individual compounds. And we do have to do some wet chemistry to prepare our samples for this kind of analysis. And then those individual compounds go into a combustion reduction reactor where they're turned into CO2 gas or N2 gas, and then that's sent to the IRMS. So those are very much in brief what the methods are and sort of how they're different.
Thank you. I think that was a very understandable explanation of something very technical. We have a question from Morgan, who asks, did historical or present-day sediment variations between sites impact preservation of fossil specimens?
Yeah, that's a really good question. In some cases, probably, but we have more work to do with that. So, a lot of these specimens are very, very recent in age. So longer term changes in sort of sedimentary deposition is not as big of a factor. What is a really big factor for us is sea level rise. So particularly in California, there's a whole series of islands off the coast of Southern California called the California Channel Islands. And several of those are now separate islands, but used to be in the late Pleistocene, early Holocene, a single humongous island. And so all of the archaeological sites that were in those areas are now underwater, right? So that presents a big issue. So I have one my colleagues, Todd Brady, who's actually on this slide, had a whole grant a few years back doing underwater archaeology, where they were sending ROVs down to the bottom of the seafloor to look for fossils, artifacts, all kinds of stuff. So unfortunately, but it's really hard to excavate at the bottom of the ocean. So it does present some challenges. So that's absolutely something we have to be aware of. It is likely that the ones that are currently underwater are the older sites, right? So again, this sort of like Pleistocene, Holocene transition. So by the time we get to the late Holocene, and right around the time Euro-Americans showed up, the sea level was about the same as it is today. So the archaeological sites that we're talking about in this talk are less likely to be affected by this, but it's definitely something we have to consider.
Thank you again, Emma. Let's see if we have any more questions. Yes. Okay, this question asks, first of all, fascinating talk. Are there or were there distinct sea otter subspecies? And if so, how has the near extinction and reintroduction affected these subspecies or the sea otter gene pool in general?
Wow, fantastic question. Yeah, so I had to gloss right over it, but the specimens we have from Oregon were actually part of a whole study on this exact topic. So let me start first with the subspecies of sea otters. So there are three currently recognized subspecies: the California species, the Alaskan subspecies, and the far northern subspecies. So there are distinct subspecies of sea otters. Generally, genomic analysis strongly suggests there has been a major population bottleneck, as you would expect, following the near extinction during the industrial era. And these Oregon samples were really interesting because one of the questions that has been asked in Oregon alongside this translocation question is, well, what subspecies should we bring? Right? Like if we're doing translocations, should we be translocating otters from the Western Aleutian Islands to Oregon, or should we be bringing sea otters from California? Or is there an entirely fourth subspecies that's now essentially extinct? So full disclosure, right? I am not a geneticist, so I don't want to try to get into the technical details and say something inaccurate. But what the data do suggest is that the specimens we have from Northern Oregon were more closely related genetically to the modern subspecies we find in Alaska than they are to California sea otters. So still a ton of work to be done. There's some awesome researchers who've contributed to this work that are very much active in sort of the sea otter genetics and bottleneck question. So it's an active area of research for sure.
Okay, great. We now actually have a follow-up question to this from Morgan, asking, was the formation of these subspecies largely driven by regional or behavioral isolation? And is interbreeding mostly in captivity, or does it also occur in wild populations?
Yeah, so it's mostly morphological and regional. So there are some body size differences, and it kind of follows what you'd expect, right? So the northern subspecies tend to be larger than the California subspecies. There are some behavioral differences, but I think a lot of those behavioral differences, so for instance, northern sea otters are a little more likely to be seen consuming fish relative to California sea otters. But I'm really hesitant to say that behavioral difference has a genetic basis. I think it has more to do with the fact that Southern California has an insane diversity of coastal macroinvertebrates because they're sort of at the edge of this cold water, warm water transition. So the diversity of potential prey items for sea otters is just greater in that region. In terms of the inbreeding, I don't have a good answer to that because that is not my area of expertise. So I will have to punt that question. But I will say that on this slide, Courtney Hoffman and Hannah Wellman have done some really excellent work on the past genetics of sea otters. So I'd recommend you take a look at their research if you're more interested in that question.
Great, thank you very much. Morgan also says thank you. We have a few more questions, if you don't mind.
Not at all.
So this one is regarding these different locations and the different food sources. So the question asks, Oregon is known for having fewer estuaries than Central California in general. Might this be the reason that ancient otters in Oregon seem to rely more on kelp-based food webs?
I think so. Again, there's more work we need to do to address that question. The one big critique we've gotten with this analysis, and it's a very reasonable critique, is that our sample size is kind of small for Northern Oregon. And in particular, right, we're looking at two sites that are very, very close together. And so I think that we really need to broaden our sampling scope to include farther down the coast of Oregon, and try to do a more comprehensive job of sampling the Oregon coast. But I do think that's what's going on. The coastline of California is more topographically complex in many ways than Northern Oregon. So I think that's a real contributing factor. The slightly confusing thing about all of this is that kelp cover in Oregon is really not that abundant today, at least not compared to some areas of California. But again, there's also lots of reasons that that could be in play, right? The removal of sea otters, for one, which are, known to sort of, through their presence and consumption of herbivorous macroinvertebrates, help promote kelp canopy recovery and kelp abundance in general. So I do think that's what's going on, but stay tuned, I guess.
Thank you very much. Then we're getting to what for now is the last question. And it's also something you already alluded to. So the question asks, you mentioned correcting for temporal, trophic, and tissue isotopic effects. Can you briefly explain how this works and which one of these factors is the most important to correct for?
Yeah, absolutely. Wow, these are all fantastic questions. Yes, so let me kind of just briefly break it down. So the temporal effect for carbon isotopes is quite important. And the reason for that is that as a result of industrial fossil fuel combustion, we have changed the isotopic composition of CO2 in the atmosphere by nearly 2 per mil over the last 100 years. So if you are not accounting for this and you are making comparisons between modern samples and historical or archaeological samples, your inferences could be off, right? So we do absolutely have to account for that. So that's a really important one. There is a great paper out by Casey Clark, and they actually developed a whole R package for doing SUS corrections in the North Pacific. So we're really fortunate to have that sort of prior study to be able to use. So we feel pretty comfortable that we corrected for those in the most robust way we possibly could. So that's sort of the first consideration. And then, because especially with nitrogen, right, I mentioned there's this stepwise increase in nitrogen isotope values as you move up food webs. So if you're trying to compare isotopic values of a consumer to potential prey, you have to account for this. So generally, what's really commonly reported in the literature is on average 3 per mil is the usual factor when you're looking at something like a muscle sample versus a dietary sample. In this case, because we're looking at bone collagen, it is a little bit different. So the nitrogen isotope trophic discrimination factor for bone collagen is a little bit different than it is for muscle tissue. But we're really fortunate to also have a data set of bone collagen, muscle, and whisker samples from the same individual sea otters. So one of my dissertation chapters, when sea otters are found stranded or dead along the coastline of the US, because they're an endangered species, you're required by law to report it. The carcasses are picked up. Typically, there's a necropsy done to assess cause of death and all that kind of stuff. And we were able to kind of piggyback off of that work and sample some of these tissues. So we are really confident in the isotopic differences between bone and muscle and whisker within the sea otter species, right? I wouldn't necessarily generalize that to all mammals, but and then there's also been a series of really excellent feeding experiments that have been done on captive sea otters comparing their whisker isotopic values to their diet. So it's a little bit of like chain of corrections, right? But because we know diet to whisker, and then we've characterized whisker to bone, we're able to correct bone to diet. And then that also feeds into this tissue-specific effect, which is really only applicable when we're comparing our modern samples to our archaeological samples. We can't sample the bone of a living animal, so we have to take a non-destructive sample, which we use whiskers. But then again, because we have this comparative data set that we've built, we're able to feel pretty good about comparing all of these very different isotopic data sets. And in terms of which one is most important, I think that's really context dependent, which I know is an unsatisfactory answer. But it really depends on whether you have different tissues or different time periods in the study that you're designing.
Thank you very much, Emma. I think it makes sense. Usually these things are context dependent. I think that was it so far for the questions. If you have another question, please type it really quickly, or just wait for the tea time. So we're gonna wait a few seconds. If there's no more questions, then I want to already thank Emma for a great talk and also a great Q&A. And also thank you to the audience for sending us great questions. And again, if there's no more questions, then I will end the Q&A session. And before we go into tea time, thank you, Emma from Real Gearty, for a great talk. And I will briefly share some information about next week's talk before we go into tea time. So again, thanks everyone for joining us. Thanks you, Emma, for the talk. Thank you, the audience, for the great questions. Again, if you would like to contribute to our demographics information, which is completely optional and anonymous, please fill out the weekly feedback form so that we can learn a bit more about who attended today's seminar. I hope that you will also join us next week on March 4, same place, same time, for a talk about vertebrate paleontology by Vanna Allroyd from Utah Valley University in the US. For those of you that won't stay for tea time, hope you have a great rest of your day and hopefully see you next week. For those of you that don't want to stay for, that do want to stay for tea time, or maybe are curious about what tea time is just a relaxed conversation about the talk with a speaker, where we turn off the recording, obviously, and everyone can turn on their cameras and microphones if they want, and just have a chat about sea otters, or maybe if you have any very in-depth questions that you would like to ask Emma, she will be available for a bit more minutes after this. But because we want to give ourselves, and especially Emma, a break, we'll first take a 2-minute break where you can just have something to drink. And then those of you that won't stay for tea time, hopefully see you next time. Those of you that do stay for tea time, see you in 2 minutes.